Our next speaker is Ilya Nemenman from Emory University.
Thank you. Thanks for having me here, for inviting me here. So what I will try to talk about is actually not about theories of the brain, but maybe in some sense, how do we actually learn theories, right? What does it mean to have a good theory, and how do you infer a theory? But the example that I will talk about actually do not relate to the actual neural computation, which is a topic of this workshop. And in fact, I could have talked about sort of different examples of brain computation, a series of brain computation. I proposed a few topic titles to the organizers. My lab generally studies things like information processing in biological systems. We try to figure out the general laws, the computational principles of that. These are just a few examples. So, for example, we have discovered that in the developing mammary ducts in mammals, there is really an information theoretic relay channel for which we can calculate the channel capacity and see that the cells actually achieve the bounds that are that information theoretic bounds that exist in this channel. We study things like information transduction in protein signaling pathways, like an FKB protein pathway, where if you cut yourself and things turn purple or pink or whatever it is, that the color that things turn, that's because of this FKB signaling system, and we can calculate the channel capacity in the system and see what limits the computation in the system. We study systems with multiple different receptors and multiple different molecules, and then on the brain side, what we try to do is we study reinforcement learning or equivalent of reinforcement learning in real animals, like for example in this worm, which learns to associate temperature and food and then makes a decision whether to go to the hot temperature or the cold temperature. And it turns out that the model that we can build sort of fit the data the way that I show, and the models are a bit more complicated than the simple reinforcement learning. They include multiple time scales and so on. Same story with birds. We study birds that learn to sing or learn to adjust their song based on perturbations, and there we realize that, for example, maybe there are different computational sort of ideas that the birds actually do compared to what we normally think they do. It turns out that their singing is very widely distributed with very, very long tails in the distribution of their songs in both pitches and amplitudes and so on. And so if you put this into the model of how the birds learn, if you don't assume that the fluctuations are small Gaussian distributed but actually do have long tails, then you can get fits to real data, how quickly the birds learn a new perturbation, which are sort of as good as this plots show, right? So we work on other things in the brain computation. So, for example, one of the last pieces of work that we have done, we have shown that the information encoding in muscles, right, in either motor neurons or in the neurons that directly synapse to our muscles, doesn't proceed by rate, at least in birds, right? Doesn't proceed by the rate of the number of spikes emitted. Doesn't even proceed by timing of individual spikes, but the information encoding is given by patterns of spikes and the timing of spikes within a multi-spike patterns of 3 or 4 spikes put together. And so this is some of the things that I can talk about, and I'm very willing to talk about them later at the end of this talk during the breaks if somebody is interested. But what I was asked to talk about by the organizing, by Wolfgang, is something else. There's different pieces of work that my group has worked on. And that work has been done primarily by Brian Daniels, who is a research faculty now at Arizona State. And the experimental data that I will show comes from a lab of Andrei Levchenko and Will Rue in Yale and Toronto, respectively. So let's start this topic with this actual meat of the talk with this short story by Borges. It's a whole short story of exactitude in science. I'm sure that many of you have seen this short story, have read this short story. In that empire, the craft of cartography attained such perfection that the map of a single province covered the space of an entire city, and the map of the empire itself an entire province. In the course of time, these extensive maps were found somehow wanting, and so the College of Cartographers evolved a map of the empire that was of the same scale as the empire and coincided with it point for point. Less attentive to the study of cartography, succeeding generations came to judge a map of such magnitude cumbersome, and not without irreverence, they abandoned it to the rigors of sun and rain. In the western desert, tattered fragments of the map are still to be found, sheltering an occasional beast to beggar, and in the whole nation, no other relic is left of the discipline of geography. Okay? And I think that this short story by Borges sort of gives us, a literary fable in some sense of what is happening with our current progress of building ever more detailed theories, ever more detailed models of various different biological systems, including the brain, right? This discussion, this sort of thought process for me started maybe at CNS meeting in 2014, where there was a very large panel discussion at the meeting, and the question that was asked there, what is going to be the final theory of the brain, right? Somebody asked that question. And the general thought at that table during that panel discussion was more or less something like this, that the final theory of the brain, the final theory of just pretty much any biological system will be a large multi-scale computational model, and what we need to do is we need to generate more and more data to fit all of the details, all of the parameters of this model, right? And I think that there is a lot of issues with this, at least when I was listening that at the time. I realized that I don't think of theories as a large computational model, right? Large multi-scale models with 1 million of parameters. First of all, I don't understand what is the meaning of a final theory, right? Is Newton's law, F equal ma and the gravitation, universal gravitation 1/r², is it the final theory of everything? Of course not, right? There are quantum mechanical corrections, there are relativistic corrections as we keep, hundreds of thousands of physicists nowadays keep on working on making even a better theory, right? Even a more accurate theory when you combine relativity and gravity and quantum mechanics together, and yet there is no success. And in some sense, we still believe that Newton's laws of gravity are a good theory, right? They're not a final theory by any means, but they're a good theory. I don't think we ever actually need a final theory of anything in either any biological system or in any physical system. There is no final theory of this cup that people have been showing here all the time, right? And the specific theory that you will have for that cup will depend on what you want to do with this cup. If you want to figure out how it breaks when you drop it, then that's a very different theory than if you want to calculate how quickly it's actually going to drop to the floor, or how it's going to heat up as you put it on the windowsill and the sun is shining on it, right? There is not a single theory that covers all of the questions that you can ask about that cup, and maybe we shouldn't be doing the same thing when we're trying to build a theory of the brain or whatever system we're trying to model. There's a very famous saying from Leo Kadanoff, one of the sort of founding fathers of modern statistical physics, don't model bulldozers with quarks. And so the idea there is that if you're trying to model large-scale behavior, like what I tried to maybe point out yesterday at the final discussion, if you're trying to model behavior of the brain, it's not entirely obvious that neurons and synapses are the right abstraction to model what the brain does and how the computation actually is happening in this brain, right? We wouldn't ever dare to model the big macroscopic object by simulating quarks and electrons and all that stuff inside that object, and yet for some reason we think that we need to be solving the brain one neuron at a time and then putting them all together and figuring out what the brain does, right? So in some sense, the idea here that theories must lose details and must be developed to explain a limited set of phenomena, right? And that's what maybe makes a good theory, is that it explains a limited set of phenomena correctly, and it throws away everything that it wasn't designed for. And so, of course, otherwise, if you want to try to model everything, then the best material model of a cat is another, or preferably the same cat, right? We don't really want to build a model of a cat on a scale of 1:1, just like I don't think we want to build a theory or model of a brain on the scale of 1:1. And I'm deliberately, for now, mixing up theories and models, and maybe we'll come back to this distinction later if I actually finish on time. So, another thing that Wolfgang asked me to talk a bit about is this question: what does it make? What makes a good theory, right? And so I thought for a bit about this a few years back, and it turns out that even in physics, we believe that we sort of understand what a good theory is when we see it, it's not a clear answer, right, what makes a good theory. So first of all, yes, everybody again knows this phrase that with 4 parameters I can fit an elephant, and with 5 I can make it wiggle its trunk, right? This was attributed to von Neumann. So this is the idea that you probably don't want to build theories on the scale of 1 to 1. And then why? Because such theories can explain everything, right? They're in some sense not falsifiable. You can just wiggle parameters by a bit, and your model is going to do whatever it is that you want this model to do, right? And so maybe what makes a good theory is that the theory is falsifiable but not yet falsified. But then how do we actually account, how do we, being quantitative scientists, how do we model, how do we explain what falsifiable and falsified is if we're trying to build a theory of something? So we have been all witnesses here of quantitative biology revolution, be this in neuroscience or systems biology or population biology. And so maybe there is also time now to start thinking about quantitative philosophy of science, right? So where we stop philosophizing but actually calculate things. And again, as I said, even in physics, where we know what theory, good theory is, the problem is pretty big, right? So this is a story from a few years back where an experiment, BICEP 2, has measured what people thought was a signature of primordial sort of certain features of inflation. And then it turned out, and then the models of inflation explain those observations. And then a few months later, some other theories proposed that those exactly same observations can be explained by cosmic dust. And then the inflationary theories were immediately adjusted to not be explaining that, to not require existence of that feature in the experiment, right? Which started a whole sort of part of process of soul searching in theoretical physics. What do we mean by having a good theory of inflation or of fundamental physics if this theory can explain both an experiment and its inverse, right? You just tweak a few parameters, and both things, essentially every experimental data is explainable by that theory. Is it then a good theory? And it's the only theory that we have. It might be that it's a true theory. It might be that it's really what's happening in the world, but it can explain everything. Is it a good theory, right? And there was a big conference of philosophers and physicists, which is where this picture is taken from, a year later somewhere in Germany, where sort of this was the mood in the room, right? Philosophers couldn't tell us what is a good theory and what is not a good theory, right? So my thought about this, and this is sort of setting up the agenda for the rest of the talk, is that maybe we should not ask philosophers to tell us what a good theory is, but instead what we should realize that most dichotomies, if you're presented with a dichotomy, a good theory or a bad theory, every dichotomy is most likely false, right? There is a gray scale of something that goes from all the way from a good theory to a very bad theory. And maybe we shouldn't be asking philosophers to define for us what that demarcation is, but rather we understand already that in order to do science anyway, we have to use statistics. Statistics is crucial for us, right? We feed the data in order to know that the data is fitted by the theory. We have to use statistics. And maybe statistics itself already tells us which theories are good and which theories are bad, because statistics can quantify, at least for example, Bayesian statistics, which I usually practice, it can already quantify for us what is falsifiable and what is falsified, right? So if I go and, let's skip this. If I go and I just try to calculate what is the probability that this theory is given by data, which is, of course, by Bayes theory proportional to the probability of receiving data from theory, and I do some kind of a Bayesian calculation, sort of Bayesian approach to this, and then all of these integrals are going to be given by a log likelihood, and some are come factors, right? And in some sense, what this means is that the first term tells us if the theory is falsified or not, how well it fits the data, right? And then the second term tells us if it's falsifiable or not. How many other data sets can it explain? And so if the same theory can explain multiple data sets, then this term is going to be pretty large, and it's not a good theory. It can explain everything that you can attach to it, right? That you can give it. And in some sense, there is not going to be a clear-cut distinction that this is a good theory, this is a bad theory. Some theories can have a larger likelihood than others, right? And this could come from the fact that they fit better, but maybe are less falsifiable. And some theories may have a larger likelihood they fit a bit worse, but they're more falsifiable. And there's going to be a balance somewhere there. But I don't think that we should be sort of defining, in some sense, that a theory that includes every single new simulation inside the brain, et cetera, is the only one that's good, or it's completely bad because it either has too many parameters, so this term is bad, or the theory is great because it can actually fit everything that you like because this term is good, right? Maybe this is an obvious thing to many of you in the room, but to me it wasn't when I started thinking about this, because immediately the next point that you can make starting from here is when you start working with theories and trying to develop a theory of a process, then you are rejecting the statement, the idea that the good theory is the theory which is a correct one, right, and a true one, right? The importance of a theory is to be able to make predictions, is to be able to generalize, is to be able to explain the data that you haven't seen, and we really don't care to a large extent whether the theory is correct or not. As long as it works, it's good enough, right? That's what those arguments will probably, statistical arguments will tell us. And if this is the case, that maybe what we should be doing is not to start modeling systems sort of by respecting biological accuracy, right? Because we already have decided that this is not the most important, or at least I've decided that this is not the most important thing, but rather try to model systems by just developing phenomenological models which are good enough to predict what the system is going to be doing, right? So you observe a few experiments, you build a model, and if your model is able to predict what's going to come next, then it's a good model of the system. If it can't, then it's a bad model of a system. And if we're doing this, then it's necessary that these models will be phenomenological, right? They will not account for every microscope. They will not be biologically accurate, or not necessarily biologically accurate. We don't care about this. This is not a criteria for finding a model. And the series would, of course, not answer every question, but they will only answer the questions for which they were designed, right? So if you ask them to explain a certain set of experimental observations, they will do it well and make predictions on similar data. But it's okay. It doesn't make a theory wrong if the theory is not able to predict totally different class of experiments, right? Newton's laws of gravitation are still a good theory, even though they are not able to tell me whether this cup is going to break when it hits the floor or not, right? So then the next question that one can ask, if we have sort of forgone this idea of biological or physical accuracy, can we, by looking at the data, just learn what an effective model of this data is? And maybe we can try to use modern computing, modern AI techniques to actually try to do something along these lines. And that's what I'm going to spend the next 15 minutes on, and show you some toy examples, and then show you maybe one or two biological examples, including behavior of C. elegans example. So what is it we're really trying to do? For simplicity, and for now we can extend this a bit, but for simplicity what we started with is we assume that the dynamics of a network, right? So the let's remove cellular here, right? The dynamics of the system, not a cellular network necessarily, but a system is temporally local, right? Whatever system I'm looking at, be it a worm, be it activity of the brain, right? And it's given, therefore, by ordinary differential equations, okay? And any temporary local dynamics we should be able to write down in terms of some set of ordinary differential equations. We're not going to fit the curves, not the dynamics itself, not the trajectories of the system. What we're going to do is we're going to try to fit the dynamics. Meaning that if I'm going to neglect stochasticity and I'm going to neglect, for now, spatial structure, I just have a few time series that I'm trying to understand, right? This becomes a problem of time series prediction. And it's a time series, maybe x1, x2. And what I'm trying to do, I'm trying to guess a system that looks like this based on a few observations of this time series, right? So I have a sequence x1 of time, x2 of time, and I'm asked to build a model that looks like this. And why is this complicated? For many reasons. First of all, we need to automatically fit these functions fi's, and how do I enumerate all possible dynamical systems, right? How do I figure out, how do I enumerate all possible right-hand sides in this equation? And then the second problem, how do I search through this list, even if I am able to enumerate this whole list, right? And then on top of it, if I have a very, very large list from which I'm searching, how do I not overfit? But that's a bit more technical. So, of course, we're not the first ones to ask this type of questions. People have been trying to do this ever since the 80s, when sort of all of the dynamical system, nonlinear dynamics, have evolved as a discipline. Most of the approach is focused on what I would call coarse graining, where you start with a microscopically accurate system, and then you start removing details and seeing what is the gist of that calculation, what is the gist of that dynamics. And I maintain that this is a good thing to do in a physical system, but not really a good thing to do in a biological system or another complex system. The reason being that in physics, we know that because of the symmetries of data, because of the symmetries of the world, there are certain sets of universality classes, which means that if you screw up your microscopic model, you will still get a correct coarse-grained model, or at least could get a correct coarse-grained models. We have no such guarantees in biology. If you start with an incorrect model of a brain with 10 to the 20 things interacting with each other, and you start removing pieces of it, if you start with an incorrect model, you will probably end up with an incorrect coarse-grained model as well. Universality is not going to save you, right? And so maybe a better approach is to do top-down, where you start with the simplest possible model and then just refine them, add additional features as needed in order to produce better and better fits rather than remove features from a detailed model. And there are multiple different approaches people have tried. The two ones that I like the most, besides what we did, is Hud-Lipson's Eureka engine, which I'm sure many of you have heard about, and also recent work by Brunton on using sparse regression to fit dynamical systems, right?
Really?
So, yes.
That's a pretty strong statement about not starting. If you start with the wrong model, you'll end up with the wrong model. And it's through
You have. I'm not saying that. I'm sorry if I said this. What I'm saying that if you started with the wrong model, you're not guaranteed. There is no reason to expect that you are going to end up with a good one, right? You might still, but there is no reason to expect that.
Expect that you won't.
That you will.
But
Right?
is there any reason to expect that you won't? But I don't know. It sounds. I'm agnostic about this. I never really thought about it that way.
So I don't know, right? I think that there are some reasons. So effectively, what you want is that as you are coarse graining, you flow towards a better model, right? The difference between a correct and an incorrect model becomes smaller and smaller and smaller. And I have been thinking about this for a couple of years now, but I haven't been able to come up with a good reason to understand under which conditions this is going to be true or not true. So how do we avoid an exhaustive search through all possible functions, which is effectively what both Brunton's and Hod-Lipson's approach do? Right? They search through literally all possible right-hand sides that you can form out of a certain set of possible elementary functions put together, and you're allowing up to maybe 20 terms in the right-hand side and so on. This becomes very, very difficult very quickly, even on big machines. So how do we avoid this? And the ideas come from really basic stuff, really basic statistics. If you're trying to fit a line right here, you don't really need to know that, even if this data comes from science, you can approximate the science with polynomials, right? X squared, x cubed, and so on and so forth. And you can choose using standard Bayesian model selection how many terms do you like to have in that fit. It doesn't matter that your original model didn't have the right primitive. It didn't have a sign in it, right? It only had the power laws, but you still will be able to produce a good fit and even reasonable extrapolation unless you are extrapolating too far out, right? So maybe we can try to do something along the same lines. The problem is that when you're trying to fit dynamical systems, when you can start with the simplest possible sort of linear dynamical system, dx by dt is some matrix times x, and then from that point on, there is two ways by which you can make your system more complex, right? Just you can be adding nonlinearities, just like in our simple example, you add x squared, and so on and so forth terms, which each one of these becomes a larger and larger tensor, or you can add hidden variables, right? And it turns out that, of course, if you are trying to do model selection in a non-nested scenario and the number of models is infinite, then there are no guarantees that you will not overfit if you even do standard Bayesian model selection. If you want to have such guarantees, then you have to be doing search through a nested family of models. And so what we propose is that basically you start with a model, the simplest linear model sits at somewhere here. You choose the most complicated model on the other end, which can fit any dynamics, including any hidden variables and so on and so forth. And then you choose a single path or arbitrary path along this line, where effectively for one nonlinearity, I add maybe one hidden variable, or for two nonlinearities, I add one hidden variable, or something like that, some weird combination. And it doesn't quite work this way, but this is the idea. And you just follow along this path, and now it becomes a nested set of models, and you choose the one that fits the data best, and this is what determines the right-hand side for you. So this is how it would work. In reality, what we do is we start with models which are almost neural networks. They're not quite right. This is a weighted sum of sigmoids rather than sigmoids of a weighted sum. And for some reason, this is easier for us to fit, which I don't quite understand why. But of course, this is the same as a neural network. This is basically dynamical equations on the inputs to a neuron rather than dynamical inputs on the voltage in the neuron, right? And so the idea is that you have your original time series dx by dt. We're going to model it with a linear relaxation, inputs from everything else, plus non-dynamical variables that can exist in data. And what you do is for every time you want to discover a hidden variable, you add one more equation here. And you couple this equation to all the previous variables. So you start with maybe, if you started with 3 variables, this would be 3 nodes, and you added the 4th, the 5th, the 10th, and they all keep coupling to the previous one, right? Another approach that works quite well, and the importance here is because of all the work that people have done in neural networks, we know that this type of models can, in principle, approximate any dynamics, right? If you make them large enough, then they will be able to approximate any dynamics, at least approximately, right?
When you add new variables, do you keep the previous differential equations constant? Or
No, we will refit. There are some, I'm obviously skipping a lot of technical things. We don't quite keep them constant. We try to start from where we were, but we allow them to change as we do the fitting and so on. Another approach, yes?
One more, I still don't get how this helps you avoid overfitting unless you're evaluating error on held out data or something.
So what we do is standard Bayesian model selection, right? So we calculate the likelihood, and then we do not the BIC penalty. That's not good enough, right? But we calculate the fluctuation determinants to the next 2 orders, to non-decreasing orders in the number of data points, which you can do with MCMC sampling. Okay. And so. The other set of models that we can build are the models which are inspired by biochemical systems, right? There's a generalized mass action kinetics where production of X is a product of functions of polynomial functions of X and the fixed variables, the inputs to the system, and then the same for the degradation. And there has been a lot of work in the late 80s on the systems of this type, and it was proven that this type of systems can actually not even approximately, but exactly fit any dynamical system with a right-hand side which is made out from an arbitrary number of combinations of linear functions, not linear, elementary functions. Right? So both of these are complete sets. I can fit any dynamics, and by adding extra equations, I have sort of set my pass through the set of all possible models. I'm not going to find the exact model. Right? I'm going to find the model that fits the data reasonably well, and let's see where it takes us. Right? So this is the first example that we tried. We generated a bunch of about 200 different fake, to simulate it, whatever, planetary motions around the sun, planets, comets, all things like that. And we tried to fit, and we just gave an equation, the time series R of t for our algorithm, and we asked it to fit this data. And what the algorithm immediately told us that it cannot do it with a single variable. It needs to have 2 dynamical variables in order to explain this, which of course is a statement that Newton's laws are second-order differential equations. There is R and there is V, and the equations that dV dt is equal to force. And then it found the force, and the force is, because the actual 1 over R squared is not within our model, exactly, it found an approximation to that force, which we can build using in this case, the S system approximation. And this is how the phase portraits of the system look like. This is a true model. This is a circle. Our model didn't quite fit the circle, but pretty much, right? This is an ellipse. This is an ellipse in our case. This is a hyperbola. This is a hyperbola in our case. The fact that the scales don't match is not a big deal for a simple reason that our model, of course, doesn't know whether you're measuring velocity in meters per second or in kilometers per second or in whatever else you wanted to measure the velocity, right? And this is now tests on held out data where we put a planet with the initial condition, initial whatever position and velocity, and the dotted lines are what this planet would actually do, and the solid lines is what our algorithm predicts for this type of planets, yes.
So how do you relate the hidden variables of the observation?
We don't here, right? And so we're working on this in 2-dimensional systems. You can. In two-dimensional systems, you can probably try to use some ideas based from more small complexes to try to understand whether these two systems, right, the true system and this one that you observed, are really the same system in some sense, right? But
my question is much more simple. So compared to your previous slide, what is x?
So x here is x is the r, and then it's r of t. So our differential equations in this case are dr by dt is equal to something, and then d hidden variable by dt is equal to something, and the fits are given by these two equations with weird polynomial nonlinear polynomial right-hand sides. Okay.
Forgive me.
Okay.
The input in the model, right?
Right.
You just keep putting in higher and higher order temporal derivatives?
No. We just give trajectories. And in fact, what was done here is actually even simpler. What we gave is the initial point for the planet, and then some random time later, what is the position of that planet? Not even the whole trajectory, but just two snapshots at different time points later.
What is the theory for how that works in general? Because obviously for Newton you need R and R dot in your set. But in general you may have a very partially observed system. Does this approach sort of still...
So let's try to I'll show you a few more examples, okay? So here is another example. I'm sorry. Okay, there's another example from, this is sort of a toy model that everybody uses nowadays in this field of automatic inference of dynamical systems. This is glycolytic oscillations in yeast, 7 species, 7 chemical species, 28 parameters, complex rational dynamical laws, right? It's a nasty system. So the way that we model the system, or what we try to do with the system, is that we say, look, suppose I only observe 3 variables out of 7, right? And we'll, on top of that, just say that there is a 10% noise for this data, and of course we explored larger noises and so on and so forth. And so the data is going to be n samples, whereas each individual sample is going to be 3 initial conditions for this chemical species, 3 final conditions for this chemical species, and the duration of the time between the initial and final. Okay? So when I say a single sample, it means there's 7 numbers, 3 in, 3 final, and the time, right? And then we have and so this is basically how it's going to look like. This is 7 species. There is some oscillatory dynamics that goes on, initial condition, initial, final, and there is total n samples like this. This is what happens when we feed this data. Now this is an out of sample, and not just out of sample. We're starting with initial conditions which were not even within the range of the ones that we've trained. We're trying to extrapolate. We're always trying to make predictions, right? And so the dotted lines are, again, what the true system would be doing, and the solid lines is what our dynamics predicts. And the reason why it's possible to do, or should be possible to do, right? We're going back to the same Taken's embedding theorem, right? If you don't need to observe every single variable, right? If you observe enough pieces along the trajectory, or observe enough similar trajectories, right? You don't even have to observe many points along one trajectory. You have a few similar initial conditions, and then you look at few outputs at different time points from the similar initial conditions. You, in principle, may be able to do these fits. There is no theorems about that, but I'm not a mathematician. I'm sort of a practical physicist in some sense, right? And so it works, right, in this case. What is even more interesting, if you notice, the original system has 7 variables. Our inferred system has 6, and this gave me a bit of sort of a worry at the first time we looked at this, but then we realized that there is a conservation law in the original system. Right? Carbon is conserved, right? Carbon just goes through all of these reactions, and in fact there's only 6 independent variables. And here it is, the system has managed to figure out that 6 variables is enough. The 3 hidden variables have nothing to do, or there are some weird combinations of the 4 other hidden variables, and I don't know what they are, and I really don't care. The point is that we're making good predictions. Okay? So now let me take you to a real data application, and I wanted to talk about 2. We'll talk about 1 because I'm running so late. So what we wanted to do to model is something where the microscopical model just doesn't exist and won't exist because the system is so complex. Can we try to figure out what the system is doing? And so here we have the C elegans. This is data from Will Rue's lab. And every time the green dot is tracking the head of C elegans, and every time the dot turns red, it means that we're hitting it with an extra infrared laser. It's hot. It doesn't like it. Rather, it doesn't like the temperature increase very, very much, and so it recoils back. And so what we try to do is we try to understand, can I model the dynamical system of this worm where the input is the temperature that it's experiencing and the output is its velocity, okay? Can I build a model and can I make some predictions that I didn't put in? The specific question that is interested people in this field is whether the two behaviors where the worm is going forward and going backwards are two independent attractors in the system, right? The forward attractor and the reverse attractor. And then by hitting a laser, by giving a laser, you are biasing the relative strengths of the two attractors, right? So the forward attractor is more stable when there is no laser light, and the backward attractor is more stable when there is laser light. Versus another possibility where there is just one attractor can continuously vary in behavior, where by increasing temperature, the worm's preference doesn't switch from forward to backwards, but sort of goes continuously from going forward with some velocity to going backwards with some velocity, right? And that's been a question for a couple of years in this C. elegans field. I'm not sure how important this is overall, but I think that, for C. elegans, but I think that the question of whether the behaviors get formed by stimuli, or whether the behaviors exist there and we just switch between them behaviors, right, is probably an important question even for larger animals like us. And so this is the snapshots of the data. This would be the lasered profiles that the worm experiences. There are three different heat intensities. I'm only showing three. In reality, we tried continuous numbers between whatever small and large. This would be the responses to these profiles averaged between a bunch of different worms. And these are now 5 different values for worms. This is what our model does, right? This is the best feed that we produce. And so the fits are reasonably good, right? The velocities that we predict are there. They're reasonably good. You can even try to make sense of this model. The idea is that, first of all, you need a second variable. The worm is not completely controlled by the temperature. It has some memory, right, which by itself is an interesting statement. This memory is suppressed by the laser current, and then there is negative feedback velocity on velocity. So the system wants to relax the velocity to a certain particular value, which is determined by the memory and the laser intensity. So you can make a story, right, why this is reasonable. And very similar wiring diagrams have existed in other systems where people looked at sort of point stimuli and the responses to this point stimuli, like, for example, E. coli chemotaxis or E. coli thermotaxis. Very similar wiring diagrams are actually molecularly present in those systems. But I don't want to really look at these equations. They are not meaningful. Why should we trust them, right? How do I know that they are correct or not? So, and do they answer the question that I asked originally? So this is the phase portrait of the system. This is the speed of the worm. This is recoil negative speed. Going forward is positive speed. And this is the hidden variable, the memory. And when you hit the laser current, hit the laser, what happens is that I keep on having a single attractor, at least in our model, a single attractor that immediately moves once the temperature raises, moves to the negative velocity, and then the worm tries to chase this attractor, right? So, and the attractor keeps on moving itself. Why? Because as the worm is moving, the temperature that the worm is experiencing is changing as well. And so in this sort of chase of attractor moving around and the worm moving around, trying to chase the attractor, you get this typical negative sort of recoil velocity. This is at least what we predict. Why should we trust this, right? Why, if I tell you that the behavior is continuously controlled, why should you trust this? And I think the test, as always in this, what I started with, test in all of this models, is whether you can make predictions about things that you didn't put in, okay? And so here we train the data on the first 1.5 seconds or so of recoil, where the worm is just going back, and our model doesn't even know, our data doesn't even know that sometime later, in about 2 seconds or so, or 2.5 seconds, the worm is going to stop and start going forward again. The velocity is going to hit 0 and go. And so let's try to predict from our model at which point the velocity is going to become 0 and the worm is going to turn and now start going forward again after the recoil. And again, that has never been put into the model in training. And I can predict this time to change to the forward motion as a function of laser intensity. And so this is the average behaviors. The dotted lines are the dots are the average behaviors from the experiment, and this is our prediction. Or I can try to predict the time of change to the forward velocity as a function of the maximum speed that the worm experiences. And so here the predictions are I would say perfect, right? This is just, there's no error bars here because there's just 1 or 2 points here, so we don't really know. And so the predictions are so good that it means that we are at least capturing some truths about the internal dynamics, or at least some properties of the dynamics, so that we can make these predictions about things that are way out into the future and which we haven't trained, right? So the model knows something. What you also see is how variable different worms are. Every dot is a worm, right? And so the part which I'm going to skip is how do we take this ideas for automatic inference and do the following thing: to model individuality by imposing that every single individual in your data set has to have the same phenomenological model, the same physiological model, yet the parameters of the models could be different, right? So you're learning a model for everybody at the same time, and then fitting the individual parameters of the model on each individual worm, or in that case, we also did it for pancreatic beta cells, because that's where a lot more money is than in the worms. And that's the part which I'm skipping for now, right? And so with that, I want to summarize that I think that when one is building models of biological systems and trying to understand them, that maybe de-emphasizing the truth of a model and instead focusing on how good is the model at predicting is at least maybe not the way of going forward, but it's an alternative way to going forward, which I personally subscribe to, and others may or may not, right? If you are doing this, then maybe it's a better thing to refine models rather than coarse-grained models, because for the same argument that we just had with Peter, it might be that coarse-graining models which are improperly specified is not going to give you a correct phenomenological model, and maybe you should start with building models from top down and add features only as necessary, right? And technically what we're doing is we're building complete and nested model families of dynamical systems. We can essentially enumerate all possible dynamical systems, or can come very close to an output of any dynamical systems with these two sets of models. And such models actually make reasonably accurate predictions, right? And so here's in my final sentence what I would like to sort of to make a distinction of why doing this and what's the difference between a model and a theory, right? And so in my mind, the difference is the following. If I go back to 17th century to Robert Hooke, who is measuring force versus extension curves in a spring, right? And so he takes a single curve, a single spring, and he plots the extension versus the force, and he gets a linear relation. And that's an interesting observation, right? It's a model that this particular spring, which is made of this particular metal, et cetera, is given by this particular model with a coefficient, Hooke's constant, that is whatever it is, right? And then you can keep on doing it on other springs. And every single time you hit it, that it's a linear dependence, but k is different. Linear dependence, but k is different, right? And at that point, you realize that maybe there is more to it than just that this particular spring is a linear spring. All springs are, right? And so at that point, it becomes not just a model, but a theory of the small displacements or small extensions of all possible springs that you're going to look at. And that's basically our goal, right? So what I'm trying to do is trying to build models of similar classes of phenomena, like in this case, worm escape, and we're looking at escape to different stimuli, and we're also looking at the same temperature escape responses in different organisms and so on. And so trying to build phenomena of models of similar classes of phenomena, and then trying to stare at them and try to see what is similar between all of these models that we have built, and trying to see, maybe all of the temperature responses in all of the animals of this size are given by this feed-forward triangle that I put on the previous slide. Or maybe not, and we'll see in a few years. Yes, please, Sophie.
I'm sorry, I missed how you actually both determine the hidden variable and the weight.
So again, you didn't miss, I didn't tell this, right? So we start with a differential system of, if I have two-dimensional, let's say one-dimensional time series. I start with a one-dimensional dynamical system, and I try to fit the dynamical system within the class of possible ones that I'm allowed to. If it doesn't work, or rather, and then I try to add another variable and try to now fit a two-dimensional dynamical system, just integrate it and try to see as an the first variable is required to feed the data, and the second one is allowed to be whatever the dynamics tells it to be. And I can try to do it with 3-dimensional, 4-dimensional, and I can do it because we're not fitting every, or not trying every dynamics, but we're trying dynamics only within a certain class of dynamics, right?
So you can basically use the fact that your trajectory are very rich to both infer x and to the parameters?
That is true. I wouldn't say that the trajectories are very rich. So in this case, what we started is, in all of these examples, we just have 2 points along the trajectory. We have multiple different initial conditions, right? So it's not a single trajectory, but there are multiple trajectories, very sparsely sampled, right? There is some trade-off. If you want to infer the dynamics and the individual parameters for each specific worm or each specific cell or whatever it is, then you will definitely want to have individual trajectories to be longer, and then many cells together or many worms together give their own trajectories. But if you want to just infer one model, then you don't even have don't need to have rich trajectories. You can just have one input, one output, and many of those.
Then you really have to restrict your dynamics a lot, no? I mean,
this is what we restricted to. We're restricted to either sigmoidal networks or the S systems, and that's it. Yes, please.
So I'm still trying to figure out what this means that you actually fit, a certain interval after the laser applied, and you can predict further out. And I'm not sure, but I think you maybe got lucky, because I mean, what could your model really make work in this domain that you have, where you have not fed in the observational data? It's the Occam term, basically. So to some extent, you were lucky because the behavior was so simple that actually the Occam term somehow led you there. But I mean, the worm could have done anything further out from the laser. It could have done a looping or something else, and then your model would have failed.
And I agree with this. I'm not going to disagree, right? And I think that this is the same story, that if you are trying to predict reasonably similar dynamics, nothing really crazy is going on, then it looks like we got it, right? If there was something really crazy going on, or looking far out, where the worm at that point is now starting to make loops, etc., like 10 seconds out, right? We wouldn't be able to predict it. But again, looking back at Newton's, gravity, the fact that it cannot predict that there exist black holes doesn't make it a bad theory, right? Every theory is designed for a certain class of conditions, right?
I don't know if this is a feature, really. I mean, to some extent, I think yes. I mean, the Occam's razor term led you in this case to doing this extrapolation of your theory.
I'm not arguing against that. I think you're right. Yes, please.
The problem that I see with fitting phenomenological models is that you might get a good prediction, but the system that you get, I mean, how easy it is it to interpret this, to get some insights from this model that you had at the end? Because that's what you're really after, right? I mean, not necessarily prediction, but also sort of insight about how the system works.
So I don't think that, for example, our approach would be very good at figuring out what's going on in detail, because we are not in microscopic mapping or whatever dynamics to microscopic variables, because we are at the outset not interested in that, right? We didn't design the system to do this. And so if that's what you're asking for, if you're trying to figure out which chemical do you need to add in order to block a certain synapse, then probably that synapse and that chemical have to be in your model, and the model cannot just glance over it, right? But if that's not what you're asking, then what we do? But that being said, sorry, one second, right? But that being said, what we have tried to do is the following. We can start with a lot of sort of microscopic models, and this we are doing mostly in systems biology context, because there are just fewer parameters to play with. So we're starting with various small microscopically accurate models, and then trying to figure out what these models would be inferred as when you do phenomenological inference on them. Sort of trying to build a dictionary between which kind of microscopic models are going to be mapped into the same phenomenological model, right? It's not one-to-one dictionary, it's many-to-one by construction, but that's what we're trying to do, at least to guide an answer to this question, rather than maybe fully give the answer to this question.
Actually, I'm not asking about more detail, I'm asking about, okay, on the level that you have matched the model, I mean, if you have 7 variables and parameters, I mean, what are the tools in the end that you have to analyze, to look at this model, and I don't know, get more out of this than just prediction? I mean, is there some insights? I mean, dominating. I mean, one insight would be that it's a 6-dimensional system and not a 7-dimensional system, right? So that would be one kind of insight. Is there more you can do? I mean, imagine complicated equations.
And we're trying to do this. It turns out, again, in 2 dimensions, you can classify every dynamical system, right? It's not hard, right? But once you start moving, and so you can answer this question, what it is that my system is doing, because you know that there is only a certain set of 2-dimensional systems, right? But in higher number of dimensions, this is a question for every that dynamical system people don't know, right? You start with a 3-dimensional dynamical system, you reparameterize the variables, and you will get a different set of equations. We don't have tools to establish similarities while these are exactly the same systems, right? We cannot even say that they're exactly the same, short of searching through all possible reparameterization of 3 variables to 3 other variables, right? And so what we are trying to do is basically do very similar thing that I mentioned a minute ago about building dictionaries, more or less, right? So we're trying to figure out what would happen to a certain class of large dimensional system, how they would be inferred in our case. If you and build basically similarity metrics between different dynamical systems based on whether the same close trajectories are mapped into the same close points at the end of the trajectory, and sort of try to see how this cones of equivalence sort of expand and so on and so forth, and build the dictionaries of that this 7-dimensional system and this 7-dimensional system means that, right? And this one means that. But again,
we should stop here.
yeah, we should stop. I'm sorry. Yes, I'm sorry.